Hola, ¿qué tal? Bienvenidos otra vez a estos Encuentros del Futuro en el marco del 7º Congreso Futuro 2018. Hoy queremos hablar de algo que no entendemos para nada, pero que es sin duda una de las grandes oportunidades del futuro, que es la medicina personalizada gracias al desarrollo de muchas tecnologías. Por un lado es el universo que nos abrió todo el desarrollo de la genómica, y por otro lado, el universo que nos está abriendo todo el espacio de los nanomateriales. Tuvimos un programa hace un tiempo sobre este tema y hoy día queremos entrar un poco aplicado a cómo esto nos puede ayudar a resolver enfermedades o cómo puede ayudarnos, por ejemplo, a regenerar, en este caso, tejidos y huesos. Para hablar de esto, hoy día tenemos dos grandes invitados. Una es Molly Stevens, ella dirige un grupo multidisciplinario de científicos que ha logrado crear ya grandes cantidades de huesos y tejidos para trasplantes y órganos vitales, así como también desarrollar versiones sintéticas de distintas nanoestructuras. Yo sé que todo esto suena a chino, pero vamos a tratar de convertirlo en español en esta entrevista. Además, estamos con Lemi Xi, él es profesor de la Escuela de Ciencias de la Vida y en el Centro del Cáncer de la Universidad de Fudan en Shanghái, China, donde fundó y dirige el Centro de Farmacogenómica. Otro gran puzzle que vamos a tratar de descifrar en esta entrevista. So, welcome you both. Thank you for being here. For us was a huge puzzle to say what you do in real life. So, thank you for being here. First of all, I would like to start asking you, what is what you do, Molly?
Okay. So firstly, thank you for having us here. It's wonderful to be here. I work on biomaterials, so I have a very interdisciplinary team. We have material scientists, engineers, chemists, surgeons, cell biologists, lots of different people. And we design materials to help the body to heal itself, but also to help us to detect diseases earlier. So it's two different fields. It's regenerative medicine and also then biosensing.
And what do you regenerate with your materials?
In my lab, we work principally on bone and heart and also cartilage. And for the early disease detection, we work on early detection of cancer, but also infectious diseases like HIV.
And this bone regeneration, for which common cases it is useful for?
You'd want to use it for situations where the bone is not repairing naturally. So bone actually is a tissue that can often repair just by itself.
Mm-hmm.
I'm sure if you had a fracture before, you'll know these tissue
Mm-hmm.
has quite a good capacity for self-repair. But sometimes if the injury is too big, or maybe if you had cancer and you had a lot of bone removed, or you need a spinal fusion operation, then the body needs a little bit of help to repair better.
And how do you help it?
We help it by developing materials onto which our body's own cells can attach, and they can grow, and they can multiply, and they can form new bone tissue. And then over time, as they form the new bone tissue, our materials will degrade away, and then in the end, you're left just with a normal, healthy bone tissue.
So it's like a seed. You plant a seed in a bone that helps you to regenerate by yourself, but you need that seed because on the contrary, you wouldn't have that.
And it's also a little bit like the scaffolding in a building. Like if you were building a building and you need the support structure. And then once you have the support structure, you can grow the bits within it.
Okay, that's pretty amazing. Please tell us, what do you do?
My job is to find the right drug at the right time, at the right dose for the right patient.
Okay,
That's what
very
we call,
specialized.
we call pharmacogenomics. It is also a multidisciplinary area where we need to know ourselves in terms of our genomics, genetics. And we also need to know how a drug interacts with our body. And we also need to know how to measure the effect or the outcome of our treatment.
And are we so different? I mean, we, as a species, as humans, we thought we shared too many things, but apparently in your work, we can discover that we are not that similar, that not always the same medicine works for all the patients.
Exactly. Actually, we have 3 billion DNA bases. However, in some cases, we only need to have one single base difference. That single base difference out of 3 billion will determine whether a drug is effective for a patient or not. So that's even though 99.9% of DNA sequence is the same, but that's only one piece of difference will determine the difference in the treatment outcome. So that's so important to know exactly what's the difference in terms of one single base difference.
Where, as a citizens, we feel like, especially with cancer, that usually doctors are just like trying to put something there that hopefully helps, but the cancer is very little at the end. I mean, there's a lot of people that dies of cancer. How could this change the cancer treatment?
Yeah, most of traditionally, I think the vast majority of cancer patients don't get benefit from chemotherapy, even targeted therapy, if we don't select, identify that subpopulation of patients who can benefit. The idea is for us to know exactly under microscope, base by base, what is that difference determine the good responders and the non-responders. That really is the key of current, what we call tailor-made medicine or personalized medicine, precision medicine. That is based on our deepest understanding of a minor difference among all the patients, even though they have been diagnosed with the same type of cancer. But genomically, they are different type of cancers.
And very clearly.
Yeah.
Let's know how developed is the thing that you are, or other things that you are doing. What's the moment right now of your work, and how could it be in 10 more years? What are you expecting from your job?
So it depends about which innovation you're thinking about. So
let's start with regenerative medicine.
So on the regenerative medicine side, both for the bone and the cardiac and the cartilage, we completed a lot of preclinical models now. And one of our materials also received some approval in Europe for human use. And then in the biosensing field, we have made materials, these point-of-care tests for use in quite a few different applications. So some of the things we're targeting straight away are things like acute pancreatitis, because there's no good test for that. And that's been tried in quite a few hundred patients
and
now.
It's very, people die of pancreatitis, no?
Yeah, and it's actually the only test that can tell you at the point of care accurately within the right clinical range that the patient has acute pancreatitis. So this has been in a few hundred patients now tested. And then the early HIV has also been tested with a lot of human samples.
Can you explain us more what is this biosensitive technology?
Yeah, so if you could visualize something a little bit like a pregnancy test, except we're doing it using a drop of blood from the patient. So you just need a very small sample from the patient, and then you process it to get out the serum. And then this can attach to the biosensing strip using some special antibodies. But the big innovation that we brought in is we developed some very small particles that can attach to the antibodies. And those particles will act as artificial enzymes. And they produce a really strong chemical signal. And so when you flow this down the strip, if you have a particular protein related, for example, to a virus, so we look at proteins from HIV virus, then this will bind to the strip, and then this big chemical reaction can go on, and you get a very dense colored line at the strip at tiny concentrations of the protein. So these biosensors we've developed that are now working with human samples are actually able to determine this concentration of this HIV viral protein at concentrations below what any other point of care lateral flow test can do currently in the world. So it's very sensitive.
And in humans, it works great?
Yeah, it works really well in human samples, as validated within Europe. And now we've set up testing facilities with our collaborators down in South Africa. And this is now getting implemented for wider scale testing in the field in South Africa.
And how do you think this could change the future?
I'm really excited about it, because if we can detect these types of diseases earlier, and I gave you the example of HIV.
We can prevent it.
Well, we can tell much earlier if the patient had it. So this is important, for example, within infants, because normally if you're testing within infants with some of the tests, the test can be very complicated because of the result from the mother is somehow clouding the issue. And also, if you can treat patients earlier, then you can stop the disease from progressing as much. So this is very important. And we're looking at this for the HIV, but we're also very interested in this for tuberculosis and, you know, a number of different test areas. So it's got widespread application. And one of the other things I talked about today as well is another different test where we look at it to actually tell us what strain of Ebola virus people have had. And this is really important just to understand the spread of infectious disease so that you can much better predict how future outbreaks are going to develop and hopefully control them better.
And why are you trying this in Africa?
One of the big advantages of our tests is that because of the special components we've used, they should be much more stable than traditional biological reagents. So functioning, for example, at much higher temperatures that you might see, extremes of temperatures or problems with storage, all those sort of things could potentially be much better with this type of new test. So it could be more robust for testing in these kind of conditions. And we're aiming, of course, for it to be cheap as well, so that we can use it.
Yeah, I was asking because this medicine looks very, very expensive.
No.
Especially for the biggest populations.
So the biosensing tests are specifically designed so that they will be cheap, so that the people that most need them will be able to access them.
Okay, let's make a pause and we return with more questions with you, okay? Vamos a hacer una pequeña pausa y volvemos con más de este tema que es como muy lejano para nosotros, pero tan importante para el futuro. Es muy interesante, pero en el fondo se me van algunas referencias. Sí, o sea, tengo que volver de todas maneras con él. Okay. Okay. Volvemos con esta conversación sobre medicina personalizada y queremos ahora hablar con Lemi Xi. What's the importance of nutrition to help prevention to prevent illnesses?
Yeah, nutrition is an important part of our body's interaction with the environment. It changes our gene expression, protein profiling, and our energy balance, and almost everything. I think it's ultimate, and also our microbiomes. So it is truly important. And there are different diets.
It doesn't matter, I mean, if you have some genetic possibility to have any illness with nutrition, could you prevent it? Or you can, what's the impact?
This is a very interesting area and quite a new area that they turn for this, called nutrigenomics. Just like pharmacogenomics. We have pharmacogenomics determines which one genomic profile should be given particular drugs. But nutrigenomics intended to prescribe the right nutrition for people with the right genomic profile. So it's a similar concept, but the evidence, based on my limited knowledge, is not as strong as in pharmacogenomics because it's much, much harder to do the study and will control these conditions, because people don't always follow your guidance, like the nutritionists give you, because it's a lot of factors difficult to be considered for. So the evidence is still being discovered, but there is an active field for people to look into the nutrition and the genomic interaction to eventually find the right nutrition to better fit the right subpopulation of people with genetic makeups.
What's the situation on this field that you work, the pharmacogenomics, right now, and what's the future, the next 10 more years?
That's a lot of development over the past few years, mainly because the rapid development, sophistication of existing technology to sequence our whole genome and to analyze the data with the supercomputers and big storage space, and the accumulation of knowledge is really at a pace we never saw before, even 10 years ago. So that made it possible. First, we can get our own genome in great detail, base by base. And secondly, we know how to analyze the data. And thirdly, we start to accumulate knowledge by having more people to participate in this kind of study. And more samples, biospecimens get collected and analyzed. And the phenotypic data, like treatment effect, get collected. Then we can start to understand what kind of genomic difference make a patient respond to the drug or not. And then this new type of knowledge, and the knowledge can benefit from new patients in the future. And we have been practicing pharmacogenomics on a daily basis in China. Not for every patient, every disease, but for cancer, for heart disease, all the drug treatment is almost, especially for lung cancer, breast cancer, it's hard to imagine we prescribe a targeted therapy without considering the genetic testing result.
Have you project we heard a lot right now, the resistance to antibiotics right now. Have you think about, I don't know if this is wrong, probably is, but have you think about the resistance of the illness to your work? I mean, how you project that relationship that maybe, I don't know, the diseases will be stronger in the future. So with this genomic, you just are developing something bigger.
Yeah, that's, if I understand your question correctly, for example, cancer, we always develop resistance, but we started to learn more and more in detail how to develop resistance. For example, cancer drug resistance and antibiotics resistance. By understanding the underlying mechanism of those resistance, provide us new ways to find new treatment approaches, maybe using regenerative medicine and nanotechnology to find new ways to treat the diseases because of their resistance.
Okay, let's dream a little bit, or not dream, probably you talk about this all the time, but how do you think would be your kind of medicine in 2050, Molly?
Oh, you start with me. I think that the sorts of interventions we can make, if we can detect disease much, much earlier, will have a huge impact on the way that infectious diseases spread, but also the way that we can treat cancer. Because if you look to diagnose a cancer much earlier, or if you can spot a recurrence much earlier, for example, if you've had your prostate removed after prostate cancer and you want to see if it's recurring, you need very, very sensitive biomarker detection. This has huge impact. I mean, all of these diseases, if you can get them much, much earlier, the prognosis for the patient is massively improved.
So we can defeat cancer, you think, in the future?
I think if we can detect it earlier, this is a major player in the battle against it, yeah.
And in 2050, what would we have, I don't know how you think if we would have another kind of, not cancer, but what do you think is going to be the illness that would kill us in that moment?
I think things like the antimicrobial resistance, I mean, these are real issues. You know, the overuse of antibiotics and how can we regulate the use much better and also stop it within, you know, farming situations and these sort of things so that we can try and prevent this antimicrobial resistance. Because if organisms and viruses and so on are constantly evolving, then, you know, that provides yet another challenge, I would say.
And how do you see 2050 in your field?
Yeah, first of all, I agree with what Molly just said. Early detection of disease, no matter infection disease or cancer, is the key for our current healthcare system. I mainly work in the cancer field, but I can tell it's truly dramatically different. If you have early detection, the patient prognosis is much, much better. And if the very late stage, no matter how hard we develop a new drug, the treatment effect will not be great. So that's to say what Molly just said is true. And from my personal view about the future of medicine, we need to know much better than about ourselves. Each of us will have, at the birth, will have our genome sequenced, and even have our microbiome sequenced. Not only once, but many, many different times, as frequently as you would like, because those change, those biomes and our epigenome, metagenome will change over time based on different environmental stress, nutrition, and drugs, and so on. So we, 20, 30 years down the road, we will know much better how our own system works and how our own biological system interact with the environmental system. And we will be better to predict how our health, our disease will behave. Then we can choose it as early as possible to intervene and to benefit ourselves.
Just to finish, because we are finishing the time, you have a multidisciplinary team, do you?
Yes.
Well, what's about this new paradigm? Like usually we thought that science was about being very specialized in something, but suddenly appears this new alternative of sharing things. How had you lived that, and how do you think will be the future for you in your fields?
Well, our group is definitely around biomaterials, let's say. In that case, it's quite specialized. But we're driven really by trying to solve real problems. And so it's a very problem-focused approach. And so you have to look at that problem and then just think of all the different parts that need to come together to solve it. It's quite different to a more traditional approach, as you say, where you might study pure physics or pure biology and look at just one pathway within those systems in a more fundamental way. For us, it's really about identifying the problem and then trying in many different ways to come together like a jigsaw puzzle.
But you learn in that paradigm, or you have to build it during the practice?
Well, it's what you need to get the problem solved. And I know that, I have my knowledge, but if I can bring on really good chemists as well and surgeons, they can contribute things that I will not know. And then together we can work as a team to do a better job.
What do you think about this?
Yeah, that's exactly what my group has been doing. I'm trained as a computational chemist. But nowadays, I work almost on a daily basis with physicians, interacting with the pathologists to identify the clinical needs, identify the right problem to work with. And I have PhD students, postdocs. Some of them pure molecular biologists. Some of them are biostatisticians. Some of them are more specialized in developing new mathematical methods, deep learning algorithms. So we have to use different fields, knowledge in different fields to solve our problem. That is, where is that biomarker that can guide us to select the right treatment for the right patient?
So the future is multidisciplinary.
Multidisciplinary.
If you want to do the sort of problem solving that we're doing, yeah. If you want to solve a fundamental equation or another fundamental problem, then maybe the future is not multidisciplinary. But for the sort of applied work we're doing, I would say, yeah.
That would be a better humanity, probably, too. So thank you very much for being here. We really appreciate your experience, and thank you for having this time with us.
Thank you for having us.
Hasta aquí llegamos con esta conversación. Tratamos de entrar en este universo que nos es totalmente desconocido, pero sin duda es el futuro. Y desde esa perspectiva tener algunas esperanzas de encontrar nuevas alternativas para enfermedades que hoy día nos traen, además de mucho dolor, muchos costos como país, como familias, mucho tiempo, ¿verdad?, dedicado a tratar de sanarnos. Así que nos vemos otra vez en Encuentros del Futuro.